Good morning, good afternoon, and good evening, everyone. And welcome to the Scientist.com Inside Scientific and Stem Cell Technologies webinar titled Fully Characterized, Standardized, Human-Induced Pluripotent Stem Cell Line and Ready-to-Use High-Quality Neural Progenitor Cells for Downstream Differentiation Applications. I'm Liam Sanyo from the events team here at Scientist.com, and I'll be your host. Today, we're being joined by Dr. Andrew Gaffney and Dr. Erin Knock from STEMCELL Technologies, who will present a standardized cell line and its differentiation into neural cells for disease modeling and assay development. And with that, I'm very pleased to welcome Dr. Andrew Gaffney. So Andrew, thanks so much for joining us today, and the floor is yours whenever you're ready.
Okay, thank you very much, and hello everyone. It's a pleasure to be with you all today. And I'd just like to take this opportunity to thank Scientist.com for the opportunity to speak on behalf of STEMCELL Technologies on our development of highly characterized induced pluripotent stem cells and neural progenitor cells for downstream differentiation applications. My name is Andrew Gaffney, Director of Stem Cell Manufacturing and Commercialization at STEMCELL Technologies, and I'm joined here today by Erin Knock, Associate Director of Neurobiology, who will be presenting in the second half of the talk. Now, before I move into our presentation today, I would like to note that STEMCELL acknowledges that our work spans many territories and that our head office is located in Vancouver, Canada, on the traditional and ceded territories of the Musqueam, Squamish, and Tsleil-Waututh nations. Now, to begin with our first part of the presentation today, I'll be taking you through our development of a highly characterized human iPSC line for stem cell research, and that's both in a traditional format as well as a single-use, high-density format to provide you with the opportunity to repeat your experiments with the same starting point of highly characterized cells every single time. Now, as I mentioned, my name is Andrew Gaffney, and I work as the Director of Stem Cell Manufacturing and Commercialization at STEMCELL Technologies in Vancouver, Canada. I completed my PhD on a split-site project at both the University of Leeds and the University of Sheffield in the UK in the area of pediatric oncology disease modeling using human embryonic stem cells. I've been with STEMCELL now for a little over 9 years, and in my time here, I've worked in field applications. I previously managed our pluripotent stem cell product portfolio, which includes teaser media for maintaining high-quality human pluripotent stem cells. Now I oversee stem cell manufacturing for the company. Now, STEMCELL is an advocate for standardizing PSC data reporting and quality control measures to limit experimental variability and to ensure that relevant and reproducible findings are shared. Our commercial iPSC quality assessments and release criteria have been developed based on recommendations and guidance from a number of different groups, and that includes the International Stem Cell Banking Initiative, or ISSCR, from the paper in 2009, the Global Alliance for iPSC Therapy, or GATE, from their paper in 2018, and the recently released ISSCR Standards document that details the minimal criteria that all human pluripotent stem cell researchers should be working to. We took on board all recommendations from ISSCR and implemented these guidelines fully into our iPSC development processes. We've also recognized that a lack of guidelines for naming PSC lines has led to confusion and duplication of cell line names in the research field. And as a result of this, we decided to use the standardized nomenclature that was established by HPSC Reg that unambiguously identifies a registered cell line, allows the tracing of subclones, and enables the assignment of different cell lines to a particular donor. So as you can see in the center of the screen, this standardized name consists of an institute code. In our case, that's SCT for STEMCELL Technologies, an I or an E, depending on if an iPSC line or an embryonic stem cell line is being registered, a donor ID, which is unique to every individual donor, and finally, a letter that designates the selected clone from that donor. Now, HPSC Reg is a global registry that aims to provide the community with a central and searchable hub for PSC lines and acts as a trustworthy data source by verifying ethical and biological conformity based on community standards. If you scan the QR code on the left or follow the URL, you'll see that this takes you to the official page on the HPSC Reg website for our cell line, SCTI003A, containing all of the information relating to donor information, ethics, derivation, culture conditions, characterization, and genotyping. Now, HPSC-Reg utilizes the APEC traffic light system, which stands for availability, pluripotency, ethics, and certified. And we've provided heightened evidence that the 3A line is pluripotent and has been donor consented with the highest of ethical standards. And because of this, HPSC-Reg has officially certified the 3A line, showing that a minimal set of ethical and scientific standards have been met. This certificate is essential for some funding agencies, including any PSC research that's funded by the European Union. So here's a table that details all of the quality control procedures that are implemented in the production of iPSC lines at STEMCELL. And as you can see, a number of cell banks are generated for each line, and that includes a master cell bank, a working cell bank, and a commercial cell bank, which is the distribution bank for iPSC vials to our customers. And in general, each of these banks are approximately 3 passages apart from each other. And all of the QC can be broken down into survival and growth that are assessed by viability and recovery assays, detection of adventitious agents by sterility testing, mycoplasma testing, and viral screening, identity testing, genomic stability and integrity by testing for the removal of residual reprogramming vector, karyotyping, 20q fish analysis, detection of CNVs by microarray analysis, and also whole exome and genome sequencing. We assess the undifferentiated state by marker expression and also pluripotency by trilineage differentiation. So here you can see the donor characteristics of the 3A cell line. The line was derived from a 48-year-old female who was clinically undiagnosed at donation. And this is one of the reasons we define this product as a healthy control. Here we present a number of different donor characteristics that are separated by those that are self-declared, such as race, ethnicity, and tobacco use, and those that we've calculated, like height, weight, ancestry, and pathogenic genetic variants. Now, the 3A cell line is optimized for maintenance in MTESA Plus on HESC qualified matrigel and is passaged using releaser. When thawing the cell line from the cryovial for the first time, we recommend resuspending the cell pellet in 1 milliliter of MTESA Plus and aliquoting the cell suspension at varying densities throughout a 6-well plate. And as you can see from the image on the right, our seeding volume recommendations range from as low as 15 microliters up to 150 microliters per well. The cell should be fed as per the recommended guidelines for MTESA Plus, and the well presenting with the optimal colony density should be selected at day 7 for passaging. It's important to note that the 3A cell line has been expanded and cryopreserved as aggregates, and we suggest that for routine maintenance, our customers continue with this passaging method to reduce the likelihood of any karyotypic abnormalities from taking over the culture. Now here you can see examples of the high quality undifferentiated cell morphology that's presented by the 3A cell line. These images were captured after thawing and expanding the cell line in MTESA Plus on matrigel. And as you can see, the large round PSC colonies presented with defined borders contained densely packed cells and developed multi-layering in the center when the culture was ready to be passaged. The iPSCs retained prominent nucleoli and presented with a high nuclear to cytoplasmic ratio, which is what is expected for high quality, undifferentiated human pluripotent stem cells. Now, as I mentioned, we recommend thawing PSCs at varying volumes, ranging from low to high in a 6-well plate. And here you can see examples of 3 separate volumes that were thawed and seeded into a plate at the same time and grown until day 7, which is the recommended time for passaging. We recommend that a culture is passaged once it's reached an optimal density, and that consists of large multi-layered colonies that appear as phase bright under a phase contrast microscope and have just begun to merge. So from the images on the screen, the culture on the right would be the most appropriate for selecting for passaging. Now here you can see a selection of some of the very extensive characterization assays that the 3A line has undergone. And this includes viability and recovery assays, sterility and identity testing, genomic integrity, including whole exome and genome sequencing, among others, assessment of pluripotency by trilineage differentiation, and the undifferentiated state by marker expression and epi-pluriscore. And that's an assay that is based on assessing DNA methylation levels at 3 different CPG sites including ANKRD46, which is methylated in undifferentiated PSCs, C14OF115, that's non-methylated in undifferentiated PSCs, and the pluripotency association gene OCT4. Now, in 2017, Florian Merkel and Kevin Egan reported that their whole exome sequencing of 257 lines of human PSCs contained numerous mutations in p53, and the proportions of p53 mutated cells increased with the number of passages in culture and rose particularly rapidly during some steps of differentiation. Now, separately to this, at the ISSCR annual meeting in 2017, Shinya Yamanaka presented findings of a point mutation in the tumor suppressor gene B-core in one iPSC line that was prepared for clinical use at Saira in Japan. And this also went on to be further described by a group at the Sanger Institute. So these findings have drawn attention to the acquisition of mutations in tumor suppressor genes in PSCs. So this was also a key focus for our analysis of this cell line. So we performed whole exome sequencing of the 3As, which allowed for the identification of genomic SNPs. And the resulting profile of genetic variants was compared against ClinVar. The statuses of p53 and B-core were interrogated in depth, and for p53 on the top of your screen here, 7 variants were detected, and for B-core there were 5. However, the good news is that they were all silent. And most importantly, no variants were identified that were previously reported by Florian Merkel, Yamanaka, and the Sanger, and no pathogenic or likely pathogenic variants were identified by ClinVar analysis. Now, it's important to identify a iPSC donor's ancestry to be able to interpret the impact of personal genetic variation on experimental results. So we process the whole exome sequencing data using EthSeq, which is an analytical pipeline for identifying conserved ancestral groups. The donor profile was compared to a reference database containing thousands of genotypes of known ancestries, and that was collated from the 1000 Genomes Project. Subsequent principal components analysis identifies donor ancestry by comparison to the reference database and calculates a percentage for each ancestry. So the resulting ancestry for the 3A donor was determined to be 78.2% European and 21.8% South Asian. Now, I should note that despite focusing on characterization and QC today, a large amount of the work that went into commercializing this cell line was to demonstrate its compatibility with a number of different differentiation protocols, some of which you can see on the screen right now. We've successfully differentiated this line into cell types of all 3 embryonic germ layers, as well as 21 other cell types, and that includes neurons, cardiomyocytes, and microglia, and also organoids, including intestinal, cerebral, and brain region-specific organoids, and they were all performed using our STEMDiff kits. As well as testing the compatibility of 3A with multiple differentiation protocols, we also tested its ability to scale up in bioreactors using teaser AOF 3D media. Now, in order to initiate this experiment, 3A cells were transitioned from 2D culture in mTeaser Plus to expansion in suspension culture for 8 serial passages in teaser AOF 3D with consistent expansion. Initially, cells were expanded using a 250 ml Nalgene storage bottle with media volumes ranging between 15 to 60 mils. The cells were further expanded for an additional 3 passages using the PBS Mini with a suspension culture volume of 100 mils. The linear log cumulative fold expansion plot indicates robust and consistent expansion with no lag or adaptation phase at the beginning of culture when the cells transition from a 2D to a 3D environment. We also saw an average daily fold expansion of 1.5, with each passage being 3 or 4 days long, and a total of 1 billion viable cells were reached after 5 passages. So I think it goes without saying that most attendees of the webinar here today appreciates that human iPSCs have emerged as a transformative tool for a wide range of biomedical research applications, and that includes disease modeling, drug discovery, and regenerative medicine. And that we also realize that with long-term culture of these cells comes some drawbacks and challenges, and that can include genetic instability, issues with reproducibility from passage to passage, and a number of labor-intensive processes as well. So Today, I'd like to introduce a new product from STEMCELL with the goal of addressing some of these challenges. And this is called iPSC Direct. Now, on the top of your screen, you'll be familiar with the traditional iPSC workflow, which involves generating a cell bank that requires characterizing prior to any expansion, long-term maintenance, which of course places selective pressure on cultures every time they're passaged, and then differentiating downstream when it's required. iPSC Direct removes any aspect of long-term maintenance, allowing you to start from the same highly qualified cells for every single one of your experiments. I should also mention that iPSC Direct has been manufactured using the SCTI 003A cell line. So the workflow is very straightforward and simply involves removing an iPSC Direct vial from the freezer and thawing it into M-Teser Plus, supplemented with Clona 2, 24 hours before initiating your downstream experiment. And this can be done on both Matrigel or laminin 521. And once the cells have recovered a day later, they are then free to be used for any application you wish, such as downstream differentiation. Now, depending on the type of assay or application that you're going to go on and perform with your cells, many are highly dependent on reaching specified starting densities. And this is of particular importance with many differentiation experiments. So to allow for this, we provide recommended seeding densities after thawing the cells, depending on if you want to reach a low, medium, or high confluency before initiating your downstream experiments. And the morphology and density that you should expect to see after 24 hours after seeding can be seen from the 3 images on the screen now. One of the key differences with iPSC Direct is that this is a single cell product, and this means that you can easily count the number of cells that you're seeding for each experiment rather than having to tediously count the averages of clump numbers. Now, as with the lower density 3A product that I presented on earlier, we've also performed the same number of characterization assays on the commercial iPSC Direct vials that you'll be purchasing into your lab. And this means that you'll be initiating all of your downstream experiments with the reassurance that your cells are of the utmost quality. And this removes one of the key pitfalls of using PSCs, and that is variability. Now, here you can see a selection of the data that we provide with the certificate of analysis for the product. And that includes things like the karyotype of the cells, copy number variants by SNP array, and the undifferentiated marker expression using OC4 and TRA160. Now, as one of the key applications of iPSC Direct will, of course, be differentiation, we've made sure to test the commercial distribution vials for their trilineage potential. And we did this using the STEMDiff trilineage differentiation kit. iPSC Direct was seeded 24 hours prior to initiating the experiment, and 5 to 7 days later, we gathered the data for the different germ layers and saw over 70% dual expression for markers of each lineage, and that included PAX6 and nestin for ectoderm, NCAM and brachyury for mesoderm, and CXCR4 and SOX17 for endoderm. And this marker expression was analyzed by flow cytometry. And as well as demonstrating trilineage differentiation using the STEMDiff trilineage kit, we also took iPSC Direct cells further by generating neural progenitor cells, ventricular cardiomyocytes, and also hepatocyte-like cells using the relevant STEMDiff kits. Now, these protocols range in duration, and the resulting cells were characterized using a range of methods, such as ICC for the neural progenitors, identification of beating monolayers for the cardiomyocytes, as well as CTNT expression, and the characteristic polygonal morphology and binucleation for hepatocyte-like cells, as well as A1AT and ALB expression. Finally, iPSC Direct also demonstrated high transfection efficiency for anyone that is interested in potentially genome editing these cells. And in this experiment, we transfected iPSC Direct with eGFP mRNA when the cells were approximately 50% confluency, and we visualized the GFP 24 hours after transfection. And as you can see from the flow plot on the right of your screen, we achieved 49% efficiency of GFP expression at this stage. And with that, I'd like to conclude my section of this presentation and to provide you with some conclusions of SCTI 003A and iPSC Direct. STEMCELL's developed the Healthy Control Human iPSC line, SCTI 003A, and that was manufactured under stringent quality control standards. The 3A line has been successfully differentiated to 24 cell types to date, That includes cardiomyocytes, neurons, and microglia, among others. iPSC Direct is a single-use, high-density version of the 3A iPSC line, and with iPSC Direct, cells can be thawed and prepared for differentiation to downstream cell types in as little as 24 hours. If you'd like to learn more about these two products, you can scan the QR codes on the bottom of your screen right now or follow the URL. Now I'll pass this presentation over to Erin, who will take you through the development of our neural progenitor cell product. So Erin, over to you.
All right. So thanks very much, Andrew, for the introduction. And in this part of the webinar today, I'm going to be talking to you about the potential of human pluripotent stem cell-derived neural progenitor cells that are derived from the 3A line that Andrew has just introduced to everyone. So my name is Erin Knock, and I'm the Associate Director of Neurobiology in the Research and Development Department here at STEMCELL Technologies. And in my past life, I led an academic career where I did my PhD in human genetics at McGill University, looking at the genetics and epigenetics of colon cancer formation, and then went on to do some postdocs at the University of Cambridge and the University of Toronto, where I really got into stem cell biology, first from a developmental biology standpoint, looking at the epigenetic regulation of neural stem cell fate decisions, and then into pluripotent stem cell biology, generating models for studying Alzheimer's disease. So the subject of this section of the webinar is neural progenitor cells, which I'm going to refer to as NPCs for the rest of the presentation. And these are multipotent stem cells of the central nervous system. They arise at the neural plate folds to create the neural tube. And you can see that demonstrated on the diagram on the left of this slide. During this time, they are specified as neurectoderm, as opposed to the neural crest cells, which you'll see here in green, that migrate out to form the neuromesodermal derivatives, and of course, the non-neural ectoderm, like the epidermis that you see marked in blue. So neural progenitor cells give rise to the neurons, astrocytes, and oligodendrocytes that make up the central nervous system. And the schematic on the right of this slide shows the progression of NPC differentiation in the developing cerebral cortex over time. And it illustrates how the NPCs transition from the left of it in a state of self-renewal, and then towards the middle, over time, primarily neurogenesis, and then as you go on in time, switching over to gliogenesis as they build up the layers and the structure of the cerebral cortex. So since NPCs are critical for building the structure of the central nervous system, they make excellent models for studying developmental mechanisms and disease phenotypes of development. So you can see overlaid on this graphic, similar to the one I showed on the previous slide, how NPC dysfunction can lead to neurodevelopmental disorders like microcephaly and lissencephaly, which can have devastating cognitive and behavioral consequences post-birth. Additionally, changes during neurogenesis could set the stage for psychiatric and neurodegenerative diseases down the line when subtle changes in the types and ratios of neurons and glia that they generate could increase the susceptibility to environmental insults and the results of aging down the line. Now, as you can probably appreciate, neural progenitor cells are being looked at very closely for cell replacement therapy and brain repair, as they have the capacity, even into adulthood, to generate new neurons and glia to replace those that are lost through disease or injury. And this graphic summarizes some of the approaches that are used to stimulate NPCs to promote either self-renewal or differentiation to downstream cell types, including things like modifying their extracellular environment, adding circulating factors to the bloodstream to induce proliferation or differentiation, or directly stimulating them within the brain to achieve those things. Finally, a more technical use for NPCs is as a highly proliferative intermediate cell type between PSCs and terminally differentiated neurons and glia. So for those people who are interested in the downstream products of NPCs rather than the NPC biology itself, they serve as an easily manipulable, editable step that can be scaled up to generate millions of neurons. And this is especially useful for drug discovery workflows, as shown here in this diagram, where hundreds of plates are needed to be able to screen potential useful therapeutics. So to generate NPCs from pluripotent stem cells, we use the STEMDiff neural induction medium with the SMAD-I supplement, and that's pictured on the right. This medium directs the differentiation of PSCs to a forebrain type NPC. And it's doing this following signaling pathways that occur during NPC differentiation in the developing brain, similar to the signaling pathways and mechanisms that I showed in some of my earlier slides when we were looking at the diagrams of the structure of the brain. And the result of this differentiation is a pure population of NPCs that express the expected markers like PAX6 and SOX1 and nestin, as shown in the picture in the middle. So we took this protocol and scaled it up using our monolayer differentiation methodology to produce the frozen NPC cell product from the STI-003 PSC line, which Andrew has just described. So in the next section, I'm going to describe the features of this frozen NPC cell product, including the marker gene expression, and protein expression, the expansion rates over passage, and the capacity of these cells to differentiate into downstream cell types. So first off, our STI-003A derived NPCs express high levels of NPC markers like PAX6, shown here in green, SOX1, shown here in red, on the left-hand side of the screen, and in the graph quantified on the right. Similarly, we also see minimal off-target spontaneous differentiation to neurons, and that's demonstrated here by beta-3 tubulin staining in purple on the bottom left. We performed bulk RNA sequencing on the NPCs, and you can see those marked in red on this heat map. And we compared the result to the parent PSC line from the STI-003A, as you can see in red on the left of this graph, and also to some of the other ES and iPS cell lines that we differentiated to NPCs using our SMAD-I kit, either via the monolayer protocol or the embryoid body protocol, as written on the bottom of the graph, as you can see. Now, across all these cell lines, what we observed was an increase in expression of a panel of expected NPC and neural genes in all of our NPC lines. And that is compared to the parent PSC lines. And you can see the names of the genes that we checked on the left-hand side of this heat map. So once we were satisfied that the NPC identity was as we expected, we examined the fold expansion of this line over 5 passages, as shown in this graph. NPCs, particularly at the beginning of development, are proliferative, and they will expand to a certain extent. And so we looked at that average fold expansion over 5 passages in culture, and you can see we saw an average of 2.8-fold expansion per passage. And so at the end of 5 passages, we were able to generate about 220 million viable NPCs from a single vial of cryopreserved PSCs. So we further examined the differentiation potential over multiple passages. And here you can see the NPCs differentiated to neurons using our STEMDiff forebrain neuron differentiation kit. And shown here are the results from freshly thawed NPCs after 7 days of differentiation and 14 days in the maturation medium. And we see efficient differentiation to neurons, as shown by beta-3 tubulin staining in purple in panel B, and low levels of off-target glial differentiation, as shown by GFAP staining in green in panel C. When we examine the differentiation potential over multiple passages post-thaw, we saw that the NPCs retain greater than 50% neuron differentiation efficiency up to passage 5, after which we saw a drop-off in neural differentiation efficiency, along with an increase in off-target glial differentiation. And you can see that as quantified in the bottom of this picture, looking at beta-3 tubulin in red for neurons and off-target differentiation to glia marked by GFAP and S100B in green. So we suspect that, similar to the in vivo timelines that I talked about earlier in this section, the NPCs after passage 5 are starting to switch from a primarily neurogenic fate to a more gliogenic fate. Now, of course, we can direct the differentiation of our NPCs to astrocytes, and we do that using our STEMDiff astrocyte differentiation and maturation kits. Shown here are representative images from freshly thawed NPCs after 3 weeks in astrocyte differentiation medium, followed by 3 weeks in astrocyte maturation medium. And we see efficient generation of S100B and GFAP positive astrocytes, as shown in green and red in panels B and C in this picture. And we looked at off-target differentiation to neurons, as shown by DCX, which is a marker of early-born neurons in panel D. And we completed a similar experiment to what we did with the frozen cells over multiple passages, looking at neuron differentiation, but this time we looked at astrocyte differentiation. So you can see the high percentage of GFAP and S100B positive cells marked in red and green directly post-thaw. And then as we increase past passage 5 to passage 6 and even up to passage 10, they retain this high level of glial differentiation. And this supports our earlier hypothesis that at later passages, these cells have a very high gliogenic potential. Now, finally, we tested the effect of multiple freeze-thaw cycles on the function of the NPCs and their ability to differentiate. And we found that the NPCs at passage 2 could withstand an additional freeze-thaw cycle and still differentiate efficiently into forebrain neurons, as marked here by beta-3 tubulin, with low levels of off-target GFAP staining in green, as you can see in these pictures. So to sum up, our STI-003 derived NPCs retain high levels of expected marker expression post-thaw. They can be reliably passaged with high neurogenic capacity up to passage 5 and high gliogenic capacity up to passage 10. The NPCs can withstand at least one additional freeze-thaw, allowing you to bank and store the NPCs for future work. And with that, we'll sum up our webinar today. We thank everyone for joining us, and we're happy to take any of your questions.
Fantastic. Thanks so much, Andrew and Erin, for the great presentations. And with that, we'll move right on to the Q&A session. So I'll invite Andrew and Erin back to the audio and video line here. All right, Andrew and Erin, you ready to answer a few questions?
Sure.
Yep.
Awesome. All right, well, we'll kick things off with a really great question here. So the SCTI003A is a genetically female line, as you said. Do you also have a plan for a genetically male line?
Yeah, we do. So we do have a genetically male line under manufacturing right now. We plan on launching this hopefully over the next 6 months or so. We hope to make many more lines in the future that are to a similar quality standard to the 3As that we just went through today. So yeah, you can look out for that in the not too distant future, hopefully.
Fantastic. Great to hear. Next one here. What is the difference between the SCTI003A and iPSC Direct?
Okay, yeah. So basically, SCTI003A is a parent cell line that we've used to make other products from. So basically, iPSC Direct is manufactured from the 3A cell line. However, that's a high density and single use version of the 3A cell line. So the difference in format between the two is if you procure the 3A cell line, this is frozen in clumps or aggregates at approximately 1 million viable cells per vial, whereas iPSC Direct is manufactured from the 3As, and we store that as single cells, so you can count exactly how many cells you're placing into each vessel that you're using at approximately 10 million viable cells per vial. So depending on what your application is, if you want to keep your cells in long-term culture, you'd buy the parent 3A cell line. If you're looking to immediately initiate downstream experiments such as differentiations or transfections immediately 24 hours after thawing your cells, we'd recommend using iPSC Direct.
Fantastic, great answer. Another good one here. So what transfection reagent is recommended? And is there a way to know the components of the STEM diff SMAD neural induction media? And also, are these differentiated cells mobile, and do they migrate when coated on ESC Matrigel?
Yeah, I can take that one.
So for transfecting the iPS cells, you could use either an electroporation system or you could use a lipid-based transfection system. The only thing that we recommend is that if you're plating cells down post-transfection, that you plate in cloner 2, because that will greatly increase the survival of your cells post-transfection. As for the components of the SMAD-I media, obviously we can't divulge our formulations, but I think it's pretty clear from the name that the main driving force behind the NIM SMAD-I media kit is the dual SMAD inhibition. So based on a lot of the literature looking at PSC to neuroectoderm differentiation, we're targeting a lot of those same pathways with the dual SMAD inhibitors that are present in the kit. And of course, the basal media is formulated just to increase the optimal health and survival of the cells during that PSC to NPC transition. And then for the last question, if they're mobile, I assume that's referring to the NPCs. I mean, you'll see a little bit of movement around, but they're not migratory by any sense of the word. So the NPCs will generally stay within a confined area. They might move around a little bit, but as they start to differentiate, they will elongate and then they will remain fixed in place.
Fantastic. Thanks a lot, Erin. All right, so another one. In the protocol of SCT, they mentioned that you can passage NPC up to 10 passages, but I usually observe abnormal morphologies starting from passage 5. Do you think that's normal?
I think it tracks along very well with what I mentioned in the latter part of the webinar, where after passage 5, the NPCs will start to transition from a neurogenic fate to a gliogenic fate. And that transition to gliogenesis is often associated with a change in morphology, where you'll see cells that were very small and phase dark and defined to becoming larger and sort of more spread out. And people, because there's a wide range of astrocyte morphologies, from something that is very kind of protoplasmic looking to something that is very spiky to radial glia that are almost neuronal in morphology. I think when people say after passage 5, I see abnormal morphology, what they're actually seeing is that range in glial morphologies that are starting to crop up as that transition from primarily neurogenesis to primarily gliogenesis is occurring. So you can continue to passage the NPCs. It's just that after passage 5, you're going to see a shift in the percentage of neurons that are being generated going down and the percentage of glia that are generated going up.
Awesome. Yeah, it makes a lot of sense. Thanks a lot. So what do you mean by a single use for the iPSC Direct?
Okay, yeah, good question. So basically, the main difference, as I mentioned, between the two products, the 3As and iPSC Direct, is the format in which we've generated the product. 3A is intended for long-term culture. You can keep the cells, going for many passages, and then pull the cells out of long-term culture whenever you wish to use them. What we mean by single use is that for iPSC Direct, you simply take a vial out of the freezer whenever you want to use it and immediately go into your downstream experiments. For this particular product, we don't permit the long-term culture of these cells unless a license fee is paid. So there is a license fee associated with the main 3A cell line. However, for iPSC Direct, there isn't, because it's not kept in long-term culture. So single use is for one particular downstream application per vial every time you freeze it.
Excellent. Yeah, that goes into a nice pair of questions that were asked here, which was whether there was license fees involved in using the 003 line and iPSC Direct, and also another one asking if there was any license fees involved in the iPSC derived NPCs.
Yeah, so basically the difference between these three products when it comes to fees, as I mentioned, is that for the 3As, there is a license fee involved. This is $5,000 a year for commercial entities. However, there is the possibility for applying for a fee waiver if you're a non-profit institute, if you're looking to purchase that line. There are no fees associated with iPSC Direct and the neural progenitor cells, just the parent line for long-term culture.
Fantastic. All right, thanks a lot. So to generate neurons from cryopreserved NPCs or neuron precursors, which one do you think would be better?
I don't think it's a question of better. I think it's a question of what do you need them to be able to do. So the advantage of starting from NPCs is that you have a little bit of expansion time before you differentiate into your neurons. So if you're looking to generate a large number of neurons and you want to thaw the NPCs and you want to expand them up to get large numbers of NPCs and then differentiate into large numbers of neurons, you can do that. Or if you have some uncertain timelines and you want to expand your NPC line and then you're thinking, oh, well, I have to go camping for this weekend, so I've got to freeze this down and maybe I'll thaw it out again and start on Monday. It gives you that kind of flexibility. Whereas neuron precursors, which are already on that path to becoming a neuron, so they're not proliferative anymore, you won't be able to expand them. You can freeze them down, but they're not going to be as robust as the NPCs when you freeze and thaw them. So you might get a slight reduction in yield, but in terms of purity and efficiency of differentiation, they're pretty equivalent.
Excellent. So for driving mature astrocytes, how many passages can you obtain from these?
I've seen that question. I'll take that question from two different angles because there's two ways that you could. So if you're thinking about how many passages of the NPCs can you derive mature astrocytes from, then that's so far up to passage 10 from the NPCs. And then if you're thinking about once you get the astrocytes, then how many times can you passage those? We've passaged them at the end of our stem-diff astrocyte maturation phase, even up to 10 passages from there. So they could go on quite a ways, but that really depends on the cell line that you're using. So if you start with the 3A derived NPCs and then make astrocytes from them, we've actually found, because of that whole switch from neurogenesis to gliogenesis, if you start with later passage NPCs, you'll get a higher rate of gliogenesis, and then those astrocytes you could even passage on 10 to 20 times.
Fantastic. Yeah, nice to hear. All right, are the non-direct SCTI-003 cells frozen as clumps or single cells?
Non-direct, so meaning the parent line. So we freeze the parent line as clumps or small aggregates at approximately 1 million viable cells per vial. When you thaw this vial for the first time in your lab, we recommend seeding at a range of densities throughout a 6-well plate and allowing that to recover for a week, and then picking the well to continue your long-term passages on from with the optimal morphology, which we look for nice clump cultures. So yeah, the parent line's clumps, iPSC direct single cells.
Excellent. And how many vials are provided when purchasing?
Yeah, so it's just the one vial. We do provide a guarantee with that individual vial that if our protocol is followed exactly upon purchasing and thawing that vial, that the cells will recover as we expect. If they don't, of course, you can get in touch with our technical support team, and we'll either look to troubleshoot or provide another vial.
Excellent. All right, what's the difference in passages between the 3A line and iPSC direct?
Yeah, so the parent 3A line, we freeze at passage 32, and iPSC direct has been frozen at passage 35.
Fantastic. All right, what do you think is the best way to get good quality NPCs from fully characterized iPSCs? And how many passages do the NPCs need to be differentiated to astrocytes? I think we covered the second part of that a little bit, but.
So obviously when you're asking us, I'm going to say that the best way to get good quality NPCs from iPSCs is the STEMDiff NIM-SMAT kit. So there are two different protocols that you can use for neural induction using the NIM-SMAT kit. There's the embryoid body protocol and the monolayer protocol. We find that the embryoid body protocol is best when you are, to start with, if you don't know the neurogenic capacity of your iPSC line. So if you are not using the 3As, if you're using something else and you don't know if it's going to make good NPCs or not, then the embryonic body protocol is really robust. It will get you some neuroectoderm and you'll have to judge the efficiency based on your starting cell line. Whereas a line that you already know is going to have a good neurogenic capacity, so it already differentiates well into MPCs and neurons like the 3As do, then the monolayer protocol is going to be a little bit technically simpler, and it tends to be more efficient time after time. So you're going to get a little bit less technical variability there. So certainty, we lean towards the embryonic body protocol, and for reproducibility, the monolayer protocol. And then, yeah, I think I said in a previous question that you can passage the MPCs up to passage 10 and still get good astrocytes from those.
Excellent. Another question about passage number. This one's a little bit different. What's the highest passage number of passages for iPSCs until they're not viable anymore? For example, could you start a differentiation protocol with iPSCs in passage 47?
Yeah, so I mean, one classic feature of iPSCs is that they can be expanded in the undifferentiated state indefinitely. So as long as the PSCs are still expanding in this undifferentiated state, we wouldn't say that they're not viable anymore. So of course, differentiation experiments could be initiated at any passage number. One risk, of course, is that with human pluripotent stem cells, there is an association between higher passage numbers and links to commonly acquired karyotypic abnormalities, which is why it's important to check these cells fairly frequently. And it's also been noted that some of these abnormalities have been linked to skewed or limited differentiation capacity. So we provide the cells, as I mentioned, at passage 32. We perform a lot of QC on these cells, which I've just been through today. So of course, after initiating these cultures, these cells would be fine to differentiate with, straight away. We've kept them going. I believe it's for about 20 passages so far in-house, and they've remained karyotypically stable. So we'd say that as long as our protocols are followed, yes, it should be fine at passage 47, but you should, of course, be checking the quality of your cells.
Perfect. All right, you recommend using the MTESR medium, but we currently use a different medium. Can we culture the iPSCs with other culture medias besides the MTESR?
Yeah, so this question is referring to our MTESR media, which is for the maintenance and expansion of undifferentiated pluripotent stem cells. So we developed the SCTI003A cell line for maintenance in MTESR Plus media. So this is the media that it was previously expanded in prior to banking. We do highly recommend that you recover pluripotent stem cells in the media of which they were maintained in prior to freezing. So our protocol for thawing states that you need to be thawing into MTESR Plus, and I would recommend you do that. If you do need to switch media downstream, we haven't done that ourselves, but I don't see any reason as to why it wouldn't work. The only thing that I would stress is that when the cells are thawed for the first time, they do still go into MTESR Plus.
Fantastic. All right, do the cells need to be plated on matrigel coated wells?
I'm not sure if that's referring to iPSCs or neural progenitor cells. For the iPSCs, I mentioned the culture system was MTESR Plus. Yes, we manufactured those cells in MTESR Plus on HESC qualified matrigel. Again, that can be switched downstream, but because it was manufactured in that system, we would recommend thawing into that system. We have also tried thawing these cells into vitronectin XF, which we also provide as well, and that did also work. So feel free to get in touch with us if you're looking for a protocol to do that. But again, I don't see any reason as to why that wouldn't work on different matrices downstream. And Erin, I don't know if you want to maybe take the neural progenitor cells, if that's what the question was about.
Sure. So our NIMS-MAT-I protocol is generally done on ES-qualified matrigel as well. But for the MPCs, if you want to thaw them and expand them, you can do that on matrigel, or you could do that on PLO laminin. And then for neuronal differentiation, we generally recommend PLO laminin or PDL laminin coating for the plates.
Excellent. All right, if I want to use transcription factor based forward programming to make neurons, should I start with iPSCs or NPSCs, do you think? And are there any advantages to use the latter over the former?
I can take that one, Andrew.
Sure.
There are protocols out there for both. Starting from NPCs is going to be a little bit, I assume that the end goal is to generate neurons from either the iPSCs or the NPCs. So the NPCs is going to be a little bit faster because obviously they've already gone through the necroderm phase. So you can generally get, with forward programming approaches, neurons from NPCs in 3 to 5 days. Whereas starting from iPSCs, it's a little bit longer. The usual protocols are sort of in the 7 to 10 days, but it's, 5 to 10 days is a pretty negligible difference. I mean, obviously, starting with iPSCs, you're going to have that almost infinite expansion beforehand that you can do. Whereas with NPCs, after passage 5, you're going to start seeing a lot more spontaneous differentiation towards the glial lineage, and then your forward programming might be a little less efficient.
Excellent. And I think in the interest of time, we'll just have one last question, but I wanted to ask you both, are there any, looking forward, are there any advancements or trends or changes that you're looking forward to in, for stem cell development or differentiation, looking forward maybe 5 years? And it would be nice to get, I think, both your input, but Andrew, maybe we could start with you.
Ooh, that's a good question. I think that moving forward into the next 5 years, it's certainly really exciting right now to see all of the clinical trials that are starting in the human pluripotent stem cell space. Most of the clinical trials up to this point have involved human embryonic stem cells, and we've seen a recent switch over to use of induced pluripotent stem cells, and of course many biotech startups and pharma now using gene-edited based approaches for creating cell therapies too. So, of course, everything we've presented today is research use only, but I think moving forward, we're seeing a large switch in this field to the clinical space and how these cells can be translated over to that space. So I'm certainly following all of the progress with the clinical trials very closely. We saw some great results that were just published yesterday from Vertex's diabetes trial, which was very encouraging. And there'll be many more coming over the next 5 years, I'm sure. So yeah, I'd say that's what I'm most excited for.
I think the thing that I'm most excited for in the neural field is how far our PSC-derived cell type differentiations are advanced. So we can make almost any cell type in the brain currently. And now it opens up a lot of interesting possibilities about what we can do when we combine those cell types together. So you'll have the ability to generate complex co-culture models with, say, neurons and astrocytes, and microglia, and then thinking forward to even blood-brain barrier modeling, where you can co-culture neurons, astrocytes, microglia, endothelial cells, and parasites. And that allows you to be able to do much more physiological modeling of diseases, of disorders of the central nervous system. And that makes that increase in the physiological relevance of these models means that we'll be able to screen more accurate therapeutics and really refine the mechanisms of disease development and progression in a better way.
Yeah, really fantastic answers. Thanks so much for sharing that. And in general, thanks so much for all your insights today. It's really been a pleasure having you both with us.
Thank you.
Yeah, thanks.
Yeah, my pleasure. And also a big thanks, of course, to the audience here for participating. And in closing, we hope you enjoyed this Scientist.com Inside Scientific and Stem Cell Technologies webinar, and we'll see you again next time. Have a great day, everyone.